Okay, I'll go ahead and start things off. So it's really my pleasure to introduce our special seminar speaker today, Olivier Pertz. Olivier is my collaborator on our Chan Zuckerberg Initiative proposal or project. Olivier did his graduate work at University of Basel before moving to Scripps to do some postdoctoral work, primarily with Klaus Hahn. And then moved on to University of Basel for a faculty position, and after a few years moved to University of Bern, where his lab is now. I really got interested in Olivier's work because I was interested in the intracellular signaling because these are major processes that control plasticity and memory. And Olivier's lab develops all sorts of amazing tools for monitoring and controlling these specific intracellular signaling pathways that are applied in a totally different domain, but are extremely familiar to me, being interested in the world of synaptic plasticity and molecular neuroscience. So we've gotten together and are starting to do hopefully some really interesting work together, and Olivier is here to visit it and move our project along, and we're really happy to hear about the ongoing work in his lab. So let's take it away.
Thank you, Kevin, for the introduction. Yeah, so all what I will show you is signaling in live cells, and maybe now to the interaction with Kevin, you might be able to do that in a brain of a living mice, which is absolutely amazing to me. So we'll see how that works. So I will tell you about how cells talk to each other, epithelial cells talk to each other with respect to signaling. And the first thing that I'm showing you is basically some epithelial cells, a really simple cell that you have in culture that you grow at confluence here on a corslate. And then you express in then a biosensor for a molecule that's called ERK. So ERK is one of the major, most studied signaling molecule out there, has been studied with hundreds of thousands of Western blots. But we measure this at the single cell level. So what you see here is the nuclei of those epithelia. And what's striking is that there's a clump of cell on the right there that has high ERK. And then there's a degradé, there's a kind of gradient of ERK activity. So when you have warm colors, that means high ERK activity. When you have cold colors, that means you have low ERK activity. And so the question is, how do cells know how to activate ERK in a clump and to do this heterogeneity? Okay? And this, until now, was widely averaged in a Western blot. Okay, and then when I show that data to some colleagues, invariably, what comes up is you need to do single RNA-seq because we are in the single cell omics era, and this is what will tell you about why some cells are on and the other cells are off. And then you can just press a button and play a movie, because we can actually make movies of those biosensors. And what I realize actually is that what happens is that the signaling is a wave. It's a wave of ERK pulses that propagate in that molar layer. So the important part is that if you have the movie, you in 10 seconds, you deterministically understand how you could have this clump of cells that were activated on a wall. And so people still think about the single cell RNA-seq, but yeah, when you do those dynamics, boom, you can understand it in a much easier fashion. And so I will bombard you with movies and telling you about which information we can get to the movies. So this is the classic way we think about MAPK signaling. So you have a cell with this network. So you have some receptors here. When you shoot at the receptor, you will activate a MAP kinase that phosphorylates a MAP kinase that phosphorylates a MAP kinase. It was torture for me when I was a student to understand why you need those three kinases. And then some transcription factors will get activated in the cell, and what you get is a fate. So MAP kinase signaling regulates a fate This fate could be proliferation, could be survival, could be differentiation. And then basically the way we think about it is we find a new molecule in the network, we perturb this molecule and we see an effect on proliferation, and we say protein A activates ERK and regulates proliferation. That's a mechanism for us. The drama a little bit is whatever you do to that system, whatever you shoot, that you shoot a growth factor, that you shoot a mechanical stimulus, that you give it some chemotherapy so that you kill the cell, in any of those cases, all those different input will activate ERK. And that's a bit the drama, is that whatever you do to the system, you activate ERK, and you don't know how ERK can regulate different things. So that's what I'm going to try to explain you. Just let me get through you, let me get with you to this pathway. So you have a wide variety of receptors. Here I showed a receptor targeting kinase, but it could be a GPCR, it could be an integrin, that to multiple molecules, as this arrow here, activate the small GTPase RAS. Again, you have the MAPK, the phosphorates the MAPK, the phosphorates the MAPK. And this leads to the activation of ERK that you can detect with a Western blot of phosphorylated ERK. And this is really, this is the bulk of the literature is done with those Western blot. And this somehow wrongly told us that ERK is on or off. But what we are doing, we have these sensors, and I'm not going to explain you how they work, but the sensors are engineered so that when ERK is off, you see a fluorescence in the nucleus, and when ERK is on, you see a fluorescence in the cytosol. When we now shoot at this receptor targeting kinase, what the sensor will tell you is that when you activate the system with growth factor, well, it's not on or off, it's a pulse. So you switch the system on, and you will switch it off in 20 minutes. Okay, so it's really a dynamic pattern. And now what you can do is you can have different receptor targeting kinase that will talk to your network. So this is one receptor targeting kinase, this is another receptor targeting kinase. So this guy will give you sustained ERK activity, and another input, another receptor targeting kinase, another growth factor will give you oscillatory MAP kinase. So somehow, depending on which button you push on the cell, which receptor or which other kind of stimulus, the MAP kinase will spit you out different dynamic patterns of ERK, and then you can ask yourself, how do those dynamic patterns of ERK emerge? And this happens because those molecules are endowed with so-called feedback loops. Here there's a negative feedback loop, so when I shoot at this growth factor, this will endow this kinase cascade with a negative feedback loop. And what happens is that I will switch the system on, but because the negative feedback loop comes with a delay, I will switch it off again. But then another receptor targeting kinase that is here will activate a set of proteins that I didn't draw here that will activate a positive feedback loop. Now, when you switch the system on, you will switch it on and it will remain on. Okay, so somehow you can think about this molecules like a little network, a signal encoding network. It's very similar to an electrical circuit, and depending on which button you press, you will endow the pathway with different feedback structures, and you will spit out different dynamics. And then there's a second network that we call the signaling decoding network that can read pulse, a sustained ERK pattern or oscillatory ERK pattern in those different fates, which are proliferation, differentiation, survival, migration. Okay, so that's cool because now we understand how with one signaling pathway we can regulate all those different fates. Okay, so what we realized, and this is really not how people think about this network. So people have thrown all kinds of technologies about the network, a lot of proteomics, transcriptomics. We are looking at the dynamics. So, and we believe dynamics is important. And so the way, so our obsession is to understand the feedback structures in this network and how when you put a different growth factor and you activate different receptor targeting kinase, you can make those dynamics. And what we realized, and did by accident, is that the way you want to do it is just not to pipette a stimulus on the cell, but is to dynamically perturb the system. So what we will do, so it's a dynamic system, and what's useful is dynamic perturbation. And the way we did that is that we had some microfluidic device that we can program by a computer that will shoot pulses of growth factors at the cells. And by the kind of experiments you can do is you shoot just 3 minutes of growth factor at the cells, and you will have a pulse, and then you add a second growth factor, and you get this ERK output, and then you try this third input regime, and you get this third dynamics. And by comparing the inputs and the output, you can understand the feedback structures that are in this in cells that are important for those for producing those dynamics. So it's not the proteomics and knowing more of the components that is important. It's playing that game of dynamically perturb the system. So we learned something with that by playing this game. So we got that published on the cover. And so by playing this game, shooting sustained growth factor or pulse growth factor at the cell, we can understand the feedback structures in the network. And the cool thing that we realized is that we can also induce synthetic signaling states, so completely new signaling states that are dynamic. And because ERK dynamics control fate decision, we could reprogram fate. And so what we were able to do is that if we put a growth factor on a specific cell type, we can, and we leave it on there, the cells will proliferate. But if we give the growth factor and we shoot it at the cells exactly every 20 minutes, then suddenly we can evoke a new ERK dynamics. And this ERK dynamics will get the cell quiescent, and we differentiate them in neuron. And that's really a proof that it's the dynamics that are important in this system. Okay, so some things that you need to know now. So the way we think about this pathways is always reductionist. So we always draw basically networks with our favorite molecules and the feedback structures, because we need something simple to think about. But the real complexity of the MAPK network is this. You have not only one MAP3K, you have 3, you have 2 MAP2K, you have 2 MAP1K, you have 20 phosphatases. So you have a huge network of proteins that regulate this pathway. And so what we wanted is to understand a bit how all those proteins regulate those dynamics. The drama a bit for that is that we couldn't do that with microfluidics, because microfluidics is not scalable. So we needed a new trick. And so that was tackled by Coralie, and Coralie published that like already 2 years ago. So the way we came up to tackle this problem is, rather than using microfluidics to shoot the growth factor on the cells, what we're going to do is we're going to use an optogenic receptor tyrosine kinase, and we're going to dynamically perturb the system by shooting pulses of light at it. And so the cool thing is you can program a microscope to do those experiments. And so what you can see here is a kind of circuit that we put in a cell. So you have a receptor tyrosine kinase that you can activate with light. Simply you put the dimerization domain on the receptor tyrosine kinase. And when you shoot light, it dimerizes, it autophosphorylates, and boom, it activates your MAPK pathway. And then we need to come up with an ERK sensor that was spectrally orthogonal to that. You can see you just shoot 100 milliseconds of blue light at those cells, and you get this beautiful pulse that you can see with the fluorescence going out and in of the nucleus. We need a nuclear marker also to track the cells. And then a postdoc in the lab came up with computer vision approaches. So we have a nuclei and we have the ERK, and we can basically segment the nuclei, put a region of interest around the nuclei. Then we can average the signals in the nuclei and in the cytosol, and for each cell, we get basically a trajectory of the ERK dynamics. So with this in hand, the first thing that you do is you do a dose response, like if you pipette a different amount of growth factors on the cell. So with the light, what you can do is you can put different light intensities and light duration. And what you can see, the more you shoot light at cell, the higher, the longer. I mean, so at the beginning you have nothing, and then very rapidly you have an ERK pulse, and then at one moment you saturate in amplitude, and your ERK pulse get longer. So really see that your system behaves in a dose-dependent manner. And then now we have the throughput, because we can program a microscope to do those experiment sequences in a 96 well plate. We have a throughput to make an siRNA screen that will to kill each of those signaling network nodes, each of those proteins. We do that in a 96 well plate. And then we put some light at the cells. Here we decided to do a sustained input of light, which will give you in some cells something that oscillates. Those are the cells that express low opto-FGFR, and in some cells very sustained ERK activity. And yeah, so you program your microscope, and overnight we do the experiment sequentially, because they are movies. So you activate, measure, go to the next one, activate, measure. And overnight you get a data set with 7,000 ERK trajectories. So people usually look in Western blots at 10 conditions at a time, and boom, suddenly we have 7,000 trajectories to look at. Okay, so it's a big data set, which is difficult to look at. Maybe what you can see from a kind of bird's eye view is that so each black traces as a cell, and the red is the average of the cell population. Maybe you can see that nothing much is happening. Okay, that's the drama in this project, is that we could torture this network as much as we could, and for most of the nodes that we destroyed, nothing would happen. And this is because, and this is known, the MAP kinase pathway is robust against perturbation. Okay, I think it's pretty unique to biological secrets. So the drama is that you have a big data set, you can look at it with the eye, but our brains are not really suitable to look at such data sets. So you need something to analyze those data sets. I'll just go to the robustness. So just here's an example of robustness. So you have your pathway that culminates in phosphorylation activation of ERK. And when you don't perturb the system, you get some cells that oscillate and some cells that do sustain. This is because they have a different amount of expression in opto-FGFR. If there's not one ERK isoform, there's actually two ERK isoforms. So if I kill one ERK isoform, almost nothing happens. If I kill the other ERK isoforms, you can see that some cells are not oscillating here anymore. Yeah, maybe a little amplitude. This is a good phenotype for us. But if I kill ERK1 and ERK2, boom, I can destroy the system. Okay, so it's rather cool, because this MAP kinase pathway is a pathway where if you have 5% remaining of ERK1 and 5% remaining of ERK2, the system works fine. So it's robust. And just to try to convince you that we did the Western blots and that we really killed those two proteins. Okay, so we had these big data sets. We did a lot of screens, and we needed a way to analyze those data sets. And I mean, in this field of looking at signaling dynamics, the bound chromatics are not as far than single-cell RNA-seq or something like this. So everybody builds its own computational pipeline. And so one way to solve the problem is just to look at the curves and to extract features about the dynamics of, I mean, what is the amplitude of the peak? What's the width of the peak? Is it oscillating? So you look at that and you try to describe your system. But the problem is that if you didn't see a phenotype, maybe you're not going to get the right feature that describes it, because we are fighting against a complex data set. And so what a PhD student in the lab, Marc-Antoine, did is that he took a convolutional neural network to analyze the data. And so what this very simple convolutional neural network does, it tries to find features in the time series data set to classify if this condition was an ERK1, an ERK2, a SOS or RAS phenotype. And so it looks at the shape of those time series data to try to classify what is the perturbation. And what it does is that it spits you out a kind of latent feature space that is a compressed version of those curves that you can then, as you do in single cell analysis, you can project in a low dimensional space. So here, by example, each point is a trajectory of a single cell. And what you can see now is that at the periphery of this cloud, you have basically cells that have curves that are different than the others. So this kind of gives you an intuition that they are phenotypes. And then the other trick that you can do is that the neural network can spit you out the trajectories, the signaling trajectories that have the most robust features that were captured to classify the phenotype. And then suddenly you can see basically some phenotypes popping up. So we saw some by eye and some others we didn't, we were not able to find. So obviously the first thing that is able to find is ERK1 plus ERK2 is able to classify that you flatten completely the pathway. Sanity checks, it works. You're feeling good. So then you have some phenotypes, which are good phenotypes. We don't really know, I cannot explain them. You have another phenotype, it's a phospholipase, and what you see, your system is oscillating a bit faster. PP2A is a major phosphatase, so I was thinking that when you take a negative regulator away, your system would be completely on, but no, it just goes up, and then it still oscillates a bit. And then we have different phenotypes. And so the cool thing with that is that we are producing more and more of those time series data sets. And with neural networks, we can just basically explore large data sets. And this only provides us with a hypothesis that we try to validate by other means after. What we learned is that 3 nodes, ERK2, CRAF, and RISC2, I'll explain them just in a minute, completely stop oscillating. That's what this analysis gave us. And when you have an oscillation in your system, it means that you have negative feedback in the system. That's known for cells. It's also known for electric circuits. And so basically, I told you that the network is robust. What we know is that network robustness emerges from negative feedback in the system. So basically, what was really interesting is that we lost oscillation, which is a signature for negative feedback when we killed this one, ERK2, RAF, and this RISC2. And what this told us is that, and this is something that the system biology people didn't know, is that there are most likely 2 different negative feedbacks loops that work in your system and are important for robustness. So it's a bit drama because MAP kinase signaling is very often mutated in cancer. And the doctors, they are trying to drug a network that is built for robustness to fight against the perturbations. So the idea is that if you kill now one of those negative feedbacks, you will be able to drug robustness and to try to shut the signaling down. And this is just to show you an example of how we did this. So we knocked down this negative feedback, RISC2, which is a protein that mediates the negative feedback in the system. And so what you can see here is basically you shoot light at the system, you activate your ERK, and then you stop your light and you stop ERK. And you can now inhibit this other node, MEK. And what you can see is that you need a huge amount of drug to really switch off this MAP kinase signaling. So at 20 micromolar, you can see some cells switch off, but some cells remain on. And when I show that to clinicians, they freak out because first they tell me, oh, it's 50 micromolar, that's huge, that will kill my patient. But they also don't have the impression because they did the Western blot, and in the Western blot was just exposed so that you saw a band in the control, and that you didn't see a band in the drug perturbed state. And you can really see when you don't put enough drug, you have one population of the cell that can resist. But now if I kill one part of the negative feedback here, you can see that I can switch off the robustness. I can drug this pathway much better. Okay. This is a completely non-intuitive result that we could get for that, and we extensively validated that in a cancer model system. Okay, so we did much more than that, but then we go for something. I will show you some other stuff. So it's the first system that allows you to do large-scale investigation of those signaling dynamics at the single cell level, because you don't need to pipette something on the cell. A microscope pipettes it for you. What we've learned, disappointingly, is that I was expecting for beautiful phenotypes. I didn't see much, but this really tells us what modelers told us before, and this network is robust against perturbation. So if you try to drug it, you won't be able to destroy it unless you identify some weak nodes or some Achilles heel. And when you drug this, you can really shut down the system. I think that's what this approach, which seems completely nerdy, brings us. So measuring those dynamics, it gives you this kind of information.
So when you observe there's these resilient phenotypes, when you knock one part of the signaling pathway out, do you see absolutely zero effect, or is there a very small, if you had it large enough in and you did sophisticated enough statistics, would you observe that the system was acting a little different, but just robust in the
Yeah, so I mean we cannot explain this, why some cells remain on and remain off. Maybe we could model it. So the simple explanation is that each cell expresses a different amount of RAS, MEG, ERK, SOS, okay? And so you have this network, and then you affect the feedback strengths.
Okay.
And so if I could do single cell RNA-seq on each of those cells and measure the amount of all those proteins, then maybe I could explain this heterogeneity. But then maybe it's also not, maybe their signaling state, what is the metabolic state of the cell, what is, here we stop the cell, so they're not in the cell cycle. So it could be multiple sources of noise that we don't understand. Yeah.
Because I see you're going to come up to this next slide anyway. What kind of cells are these? And I guess why are they responsive to light? If they're like, I wouldn't expect them to be if they're inside the body.
Yeah, no, I mean, so those are fibroblasts, okay, and they are responsive to light because they express an optogenic receptor tyrosine kinase. Okay, so we engineered a receptor to be sensitive to light to activate this MAPK pathway.
Okay.
Okay, so we don't care about the cell too much here. Okay, it's just the way you can think about it is we just use the cell to build a circuit to study.
Basically adding energy to the cell, the system, in order to get it.
Yeah.
Right. So, okay.
Okay, so you've seen that we have a little obsession to hit on the cell with a hammer. It's like macrophysics, you shoot growth factors at the cells, or it's the light. But what is clear is that the cells don't see a full amount of growth factor. Just people think about that because they pipetted something because it's easy to do. Okay, cells, they basically sense chemokine gradients. We can also think of the cells. Maybe the cells gets 3 different growth factors at the same time in gradients and needs to respond to this one and not to the other one. And so we need more subtle perturbations. So maybe we need just to shoot light at one place to mimic a gradient. And so this student here, Lucien, he built a so-called real-time feedback microscopy. So what you do is that you use a little mirror, digital micro mirror, to just shine light exactly where you want it on the cells to the optics. And then what we do is we do the images in real time. So the object will move, and we can update where we're going to shoot light at the cell. It's going to get clear why this is important. So what you have here is that we trained the microscope to always shoot light at the front of the cell, and now we have a remote control. So while the cell is moving, because we do the images in real time, we are updating the light to be shooting at the top. And with this, we have a complete remote control. We can completely steer the migration of those cells. So this mimics a cell that saw some kind of a gradient. Okay, and yeah, the cool thing is that now we can repeat that on a screen. And maybe we see new phenotypes also, maybe for directional migration, because all this machinery is important for this directional movement. We wanted to take it a step further. So the cool thing about this pipeline is that you have optomic actuators, so you don't need to pipette again something at the cell. You can now even shoot light wherever you want on the cell. And we can directly measure the ERK output because we have the sensor. And we reason, so here, for example, it's kind of interesting. This cell was a very adherent cell, and we tried to shoot light at it, but it didn't want to move. Some cells that are more slender, we move with the light perturbation, but this guy didn't want, so there's some heterogeneity that we want to understand. But what we would like to do is that we would like to identify a kind of light input, spatial light input that makes our cell move really directionally, and we expect that this will tell us something about the spatial organization of the signaling in the cell. And so obviously what you could do now, we have the throughput, we can do a grid screen where we shoot a gradient of light at the cell, just the light at the front, different shapes. Then we can play with the frequency of how we shoot the light at the cell, or we can play with the intensity. So we could do a grid screen on the experiment because everything is beautifully automatized. But there's a large parameter space, and so the thing that we have the first proof of concept that this is valuable is we want to apply a machine learning paradigm that's called reinforcement learning. So reinforcement learning is an algorithm that can, for example, learn to play entire breakout. So what it will do is it will play the game, assess if it did a good score or not, and then it will continue to explore parameter spaces where you got a good, where the reinforcement learning algorithm got a good score. And for this, it can kind of in a Bayesian way try to explore a parameter space to learn the rules of the video game. And the long-term ambition is to do a video game with the cells. So if we see, by example, that this light shape makes our cell move a little bit, not good, but this pattern of light makes the cell move better, and that's a good space to explore. And then with this, you're able to basically learn really the best input of light to make your cell move. And with this, you can learn something about how this is spatially organized. So here we have a small reward, and here we have a big reward. And we hope that this will allow us to explore kind of emergent behavior in those signaling networks. So we're at the beginning of that, but we have the first data set that these approaches work. Okay, so now I'm going to switch gears. It was more slowly than I was thinking, but yeah, it's okay. So for now, you have understood the obsession is to understand how, when you put a specific input, the growth factor mechanics, stress on the cells, you induce a dynamic pattern of cells, and how this is then decoded in a fate decision, again, proliferation, survival. Now, one thing that is where it might be interesting is in setting where you have a collective behavior of cells, where multiple cells, and where those cells have to talk to each other to coordinate their fate. And we're gonna concentrate on epithelia, so your skin. The job of your skin is to maintain the same density of cells so that the skin, the epithelium, can work as a barrier. This is here. At the same time, this is your most stressed organ in your body. So your UV scratches, your cells will die constantly. And so the way people think about the epithelium is like a kind of ecosystem where the cells will talk to each other. And if 10 cells died, the cells can somehow deal with that and say, hey, 10 cells died. Please proliferate so that we have enough cells. Okay, this is called epithelial homeostasis. I will give you some idea how the cells can do this. This is work from Paulo that was published, and that's what Swiss postdocs do in Switzerland.
He's on the front of the bus.
Exactly. Yeah. Thank you. And so this started all by an accident. So basically what you see some breast epithelial cells with again the same MAPK sensor. And here you have them in response to a growth factor. So in conditions where the cells are growing. And maybe what you can see is that when I color code the ERK, basically what you have this stochastic pulsing. So in each cell, they shoot a pattern that looks random, and what's interesting is that they shoot pulses that have always the same amplitude and pretty much the same width. And it's not only about ERK pulses, we also put another Bauer sensor for AKT. AKT is another major regulator downstream of the receptor tyrosine kinase. So when you have a growth factor on the cells, you shoot, and what's interesting is that when you put more and more growth factor on the cells, you shoot with the same amplitude, but a higher frequency, and somehow the frequency tells you how efficiently you go in the cell cycle. So basically, the system that reads, that converts the concentration of growth factor into a cell proliferation unit is digital. It encodes a frequency of ERK pulses that tells you if you go in the cell cycle. And the very simple experiment we did is that we took the growth factor away to see what happened, and we expected to see no pulses. But what we saw a completely new behavior happening. So you still have the pulses, but they are less frequent. They are less frequent. But suddenly what we saw is that suddenly we saw some kind of collective behavior where cells were somehow shooting collectively as a kind of mini wave. So we had this result. So what you do, the first thing is you look very carefully at your movies. And what we realize actually is that when we take a growth factor away from the cells, they are not happy, they start to die. And what we realize is that just when a cell dies, and you can see that when the cell dies, it squeezes and gets kind of squeezed out of the epithelium. Just when the cell dies, boom, you have a wave of ERK activity that comes out at the place where the cell died. I'll tell you just, and this is 3 figures in the paper, what that means. So basically, when a cell dies, it will make a wave of ERK and AKT pulses, a wave of ERK pulses in the cells that are around. And those cells that receive the wave, that receive the pulse to the wave, their job is that they will survive for 4 hours. Okay, so it's very simple. When a cell dies, it sends a wave and it says to the cells around, please don't die in the next 4 hours. That's the time we need to repair the epithelium. And so a very simple way to explain how the epithelium engineers a system so that there's not 2 cells that die at the same place, because then you have a hole in your epithelium. So that's the function of the wave. The question is how the waves are regulated. So what we realize is that when the cell dies, they activate a so-called MMP, MMP is a matrix metalloprotease. So this is something that will cut a ligand. So EGF is a ligand that will activate the receptor tyrosine kinase. So they cut the ligand that is expressed at the surface of the cells and will activate this receptor tyrosine kinase that I showed you in the first system to give you a pulse of ERK and AKT activity. So that's how it works. And now we have a system for how the dead cell activates the next cell. So again, when this cell dies, it activates the MMPs to cut the growth factor that activates the MAPK kinase pathway in the next cell. And what we realized, and us and other people, is that when you activate the MAPK kinase pathway, actually what you do is you activate the cytoscan and the cell will contract. So basically a pulse of ERK gives you a pulse of contraction, and this contraction, we don't know exactly how it works, will then activate this MMP system and activate the next cell. So what's important is that it's not a wave where the cell shoots out the growth factor, diffuses out. It's a trigger wave where one cell activates the next cell, activates the next cell. And in there, there's a mechanical input, and we think that the mechanical input to waves through transmission that explains why the wave stops at a certain time. Okay, so that's how waves emerge. Another important aspect to the system, I told you, when you have growth factors, you have this stochastic pulsing. When you have, when the cells start to die, they make the wave. And the system is there to spatially control those ERK pulses. And what happens is that the cell reads the amount of ERK pulses to decide if it proliferates, if it survives, or if it dies. And obviously, when you put a growth factor, you have this stochastic pulsing, you have a high ERK pulse frequency. And when you have this, you have local survival, and when the cells don't receive any pulse, they will die. So the logic of the system is to spatially basically control the ERK pulse frequency, and the ERK pulse frequency tells you if you proliferate, survive, or die. And again, this explains you how one MAPK kinase can regulate so many different things. Can we formally show that ERK pulse frequency regulates fate decisions? Oh, I lost that slide in the meantime, sorry, so this is gone. But what we can do is we can use optogenetics, and we can reprogram the cells with specific frequencies of ERK pulses, and we can show that we can control proliferation, survival, and death. So the take home is simply, this is a very simple system where when a cell dies, it activates pulses of ERK and PI3 kinase signaling around the lesion, and it just buys 4 hours of survival so that there's enough time that this lesion will be repaired. That's what happens at the scale of the cell. So spatial distribution of apoptosis. But a very simple emergent property of the system is that if you have a little amount of cells are dying, you have a little amount of survival. If I now put chemotherapy on the cells, I will kill a lot of cells, and I will have a lot of waves. And the cool thing with this very simple system is that you can adapt to the amplitude of the stress that kills the cell. And I think the beauty of this is that it's a message that you can immediately capture by intuition, not some kind of omics where, yeah, I don't know, this phosphocytes regulate survival or something like this. Okay, so that's really the beauty of looking at those dynamics. And we've really shown that if you shoot a lot of chemotherapy at the epithelium will survive while it's being stressed. If we disable this wave system, the epithelium will disintegrate. Okay? So that really explains us how we are robust against chemotherapy drugs. Okay, so another stuff that is important is that we see waves everywhere in our data sets, so in cancer, in development, in stem cells, and so there was a need to basically analyze automatically those data sets, and those guys here in my lab, they took actually some algorithm that is used for weather prediction track those waves, and now we can basically compute all the features of those waves completely automatically. And I'm already a bit too late. I will just show you one movie. So we can do this trick in 3D organoids too, for the ERK-KTR. And this is because Andy, who is here, with whom I had a grant from Drexel University, was able to build a computer vision pipeline that allows to analyze this in 3D, which is much more complicated than in 2D. And I'm not going to get you to this system, which is a bit complicated. Slowly stop here. So the take home, okay, is that if you want to understand those signaling systems, so most of people have done Western blots. So obviously, it's immediately intuitive that if you do a Western blot on cells that do waves, you average out this collective behavior. So to understand this system, where input encodes different dynamics. So one trick that I showed you is that you need to measure dynamics. What's good is if you dynamically perturb the system. And so we are obsessed about understanding these feedback loops and how they spit out those dynamics. And we have other projects using the same technologies aimed at understanding how a pulse or sustained ERK pattern is decoded in the transcriptional programs that will give you proliferation, survival, differentiation. So that's one level. So really at the single cell level and at the dynamics. And then what's kind of interesting is that when you put a lot of cells together with very simple rules, these ERK waves, you can make the cell talk to each other and somehow do division labor. And this explains how a whole tissue and the cells of a whole tissue can talk to each other, and the whole tissue can respond to an insult or to a growth factor. Just one last slide is this is the latest baby of the lab. So here in epithelial cells, we have those waves. The job of the waves is to count the number of dead cells and to adapt with survival or proliferative signaling. If we now put the ERK sensor in stem cells, and those stem cells can give you basically can differentiate in different developmental phases. So mesoderm, endoderm, ectoderm. What we see is that the ERK, the logic of the ERK signaling is not to do waves, but is to do a kind of band of cells that have the same signaling with a sharp boundary of cells that don't have the signaling. And so basically what will happen is this is a kind of patterning that will tell you where you will position your ectoderm, mesoderm, or endoderm during differentiation. So I think for me it's fascinating because you have the same signaling pathway and you endow it with some different feedback loops and different ways that the cells talk to each other. You can get waves of pulses or basically patterns, long-range patterns of something that is sustained. And again, it's using the dynamics imaging that you will understand this, because this was until now averaged by Western blot. So I will stop here. This is the lab, the latest lab outing. That's the kind of stuff we do in Switzerland. Fantastic team. I'm really grateful to my funding sources and many collaborators, and Andy really made the image analysis of the 3D data set possible, something we could not put up, we could not do in our lab. So really grateful for that. And yeah, thank you for your time.
One of the things I'm learning about ERK is how important it is in a lot of different biological systems. So, for example, the retina. Do you see adoption of, and your approach has really changed the way people have looked at ERK for the last 50 years. Do you see that being adopted more widely? Or are we gonna have to wait for the old biologists to die off before people start using this?
Yeah, I like the idea of the old biologists die off because they are sometimes stubborn. No, I mean, I think this. I mean, so I was just at a conference where everybody was talking about signaling dynamics, so P53, MAP kinase, PI3 kinase, NF-kappa B, YAPTA. So this community is coming up. The tools are coming up. You realize that we are generating big data, that we need approaches to analyze big data. One big thing that came is collective behavior. Okay, so what's kind of interesting is that it's ecologists that worked a lot on collective behavior and how a flock of birds or a school of fish work together. And yeah, this community is being there, and the tools are easier to use. The computer vision is getting easier and easier to use by example, by this Napari plugin, which is the new image shape for that is compatible with Python. So yeah, I think it's a technological advancement that, yeah, there is really a committee that matures and that will look at that. And I will tell them to focus on the retina first.
So the idea of the robustness of the system due to the multiple feedback pathways are interesting, but it also seems counterintuitive in the way that, you know, if the cell itself wants to change its behavior, it then also has to change all its negative feedback pathways, because if it only were to manipulate one arm, it wouldn't do much. So you know, what kind of balance does the cell strike?
So, I mean, yeah, very good question. So, I mean, there's a trade-off for robustness. It seems to pay if you want to do different things. So, for example, in the last stem cell data set, it's not pulses. It's very sustained signaling, and it's sustained on or sustained off. And we know that the negative feedback in that system is extremely low. We still know that there's two negative feedback. So, yeah, so maybe one question to ask is, do you need more robustness if your system is frequency modulated versus on and off? So what we also don't know is how the cells decode those dynamic signaling pathways. So yeah, I mean, very good comment. We don't know yet. We've been thinking about that. Yeah, I think modelers are talking about that. Experiments are not existing yet, but yeah, that's something we need to think about. Yeah.
And along similar lines, you know, what sets the theoreticity in the cell? What controls the oscillation frequency?
Yeah. So, you know, the epithelial cell, it's not an oscillatory system, it's an excitable system. Okay, so so we see some oscillatory behavior, and this you can beautifully capture by modeling. That's not a problem, but so again, in this excitable behavior in the epithelium, the frequency has not an oscillation. It's really so it really emerges through interaction of the cells. So one easy way, so one very simple way is this wave. Okay, so you can really think it's very simple. Okay, if you put a lot of chemotherapy, there's going to be a lot of waves, and then you simply have high frequency of signals in the cells around the cells that are dying. But when you put the growth factor, most likely there's a deterministic reason why one cell shoots at a specific time. We don't know yet. Okay, yeah.
I have a question about the way the waves are propagating. You mentioned that there's the matrix metalloprotease part of the signal, and there's this mechanotransduction sort of signal. So are do both of those things happen together to signal to the next cell over?
Exactly.
And then do you know how the mechanical signal is sensed?
Yeah. So I mean, we don't know how the so the first thing. So I get to tell you again how it works. So one ERK will actually phosphorate myosin light chain, and when you have a pulse of ERK, you see a pulse of myosin activity, so a pulse of contraction. One piece of the puzzle that we don't know how it works is how the mechanics is somehow activating the MMP to clip the EGF that activates the next cell. Okay, so this we don't know, but there is, yeah, this was a super difficult experiment because you need to do biochemistry in one cell. Okay, but there is reports of mechanosensitive MMPs. Okay. And then after, what's kind of interesting is that if we know very well that different epithelia are better connected mechanically than the others. So there's these famous MDCK cells. Yeah, they make a perfect epithelium that is really mechanically connected, and they make huge waves. There's MCF10A cells, they do a kind of loosely connected mechanical object and make small waves. So this is again another kind of way to how to understand mechanics. And what's interesting now in the stem cells, ERK activity will be activated. The ERK activity is sustained, and what we see is that the contractility is sustained. And so the logic of in the stem cells is when you specify your ecto, endo, and mesoderm during gastrulation. What the cool thing is that what the ERK does here is that it contracts locally the tissue that is differentiating, so that you have this during gastrulation. At one moment you have a disc of cells, and you contract it, and it bulges, and that's how you make mesoderm and ectoderm. So, yeah, the connection to the mechanics is absolutely clear. But we don't know the details. Yeah. Question there, yeah.
Question from the chat. Dr. Wang, could you give more details on the MAPK sensor?
On the what?
MAPK.
MAPK.
MAPK sensor. Okay, yeah. So the sensor of maybe the first thing to say is that it's the second signaling pathway that has seen the most engineering in biosensors. Obviously, the first biosensor that has been most engineering is a calcium sensor, because the neurobiologists are so interested in it. Okay, so and then just after the calcium sensor, there's the MAPK pathway sensor. We have two sensors that we use. One is a FRET sensor, where you have a substrate and a little domain that binds to the substrate that is phosphorylated. When this happens, two fluorophores that do FRET come together, and you can measure the FRET because the fluorophores come together. And then there's this ERK-KTR, which is basically MAP kinase substrate that is sandwiched between a nuclear localization sequence and a nuclear export signal. And so the idea is that when you phosphorylate that, you change the local the charge of the peptide between those two signals, and you favor nuclear localization versus nuclear export. That's the two tricks that you can use to build one of those sensors.